Welcome, Biochemistry 1 class, to our very first episode of
Chemistry with Varghese and Asmus. I'm John Asmus.
And I'm Bennet Varghese, and we're here to talk to you guys about the amazing enzyme Chymotrypsin-C. And my boy John is about to give us a quick introduction about this amazing enzyme.
Thanks, Bennett. So Chymotrypsin-C, also known as kallikrein, is a member of the peptidase S1 family. So what this means is that it is a serine protease that cuts peptide bonds within polypeptide sequences. And it's actually found within us. And so it's found in our digestive tract, specifically in the duodenum of the small intestine. And it's really important with digestive and excretion processes in the body. So next, I'm going to let Bennett tell you a little bit about the sequence.
Thanks, John. This is a big ask to give me, but hey, I think it's crazy that it has 268 amino acids, 804 bases to be exact, but there's only 3 active sites, and that's all it needs to do all the great stuff that it does for our digestive system. To highlight the specific active sites, we have histidine, serine, and aspartate. I mean, that's all it is. And another thing that I want to highlight in this is that there's so many hydrophobic amino acids. And one thing that John's going to talk about is a hydrophobic pocket that this creates, which makes it very attractive to hydrophobic amino acids. I just think that's really interesting. John, do you want to talk to us about the structure for Chymotrypsin-C?
All right, yeah, so the structure is, here you can see it's a globular protein, and it has many different secondary structures. So all these sheets here, you can see these are the beta strands, these little flat boys here, and there's going to be 16 of them. And then there are 5 helices. You can see an example of one down here. And there are going to be 2 terns, which I think you can see down here, this little purple strand. But the most important thing is going to be that has a substrate binding pocket that has 3 active sites. And so that is where the polypeptide sequence comes in, and that's where it's going to bind to it and make all the changes there. And so next, Bennett, do you want to talk about the function?
Hey, if you say so, I'll take it. Now we're going to get a refresher of what an endopeptidase is. And I'm sure you guys know, but I'm just going to refresh your memory a bit. It goes inside of the amino acid sequence, not the outside, the inside. So it goes right into the middle and breaks it up. And so what that means is that it's literally what I just said. It'll cleave the inside of the amino acid sequence. And again, what John was alluding to with the binding pocket, it's a hydrophobic binding pocket. And what makes chymotrypsin in general so important is that it allows aromatic and other very hydrophobic amino acids to combine and get cleaved. And that's not what a lot of enzymes can do. It's a very hydrophobic enzyme. So that allows those hydrophobic, aromatic, whatever, super hydrophobic amino acids to get cleaved and bind. So to be specific, we got leucine, tyrosine, phenylalanine, methionine, tryptophan, glutamine, and asparagine. And again, it's zymogen activator and deactivator, meaning that it leads to other cascades of other enzymes, and it leads to that, and it can also stop it. John is going to have the really difficult task of talking to us about what the mechanism is, but he's a smart guy, so I completely trust him. John, do you want to take it away?
I don't know about that, but I'll see what I can do. So first, what we're going to have is these hydrophobic residues on the polypeptide chains come. They're going to bond inside of this hydrophobic pocket that you can see right here that I'm circling with my mouse. And so, So what this is going to do is they're going to non-covalently bond in this pocket here. And then here you can see the 3 active sites. You have aspartate, histidine, and serine. So the first thing that's going to happen here is you're going to create a first transition state. What's going to happen is this hydrogen you can see over here that is right now was originally bound to the serine is going to transfer over to the histidine, and that is going to allow this double bond from the oxygen here to come down and form a bond between the serine and this carbon right here. And the next step in that is going to be the cleavage of this peptide of the polypeptide strand into 2 different strands. So that's the way it's going to happen, is that you're going to have the hydrogen that went to the histidine is now going to come over and bind to this nitrogen here, and that will cause the cleavage of this C-N bond right here. So now you're going to have 2 strands, and this one down here is going to be a free strand now, but this strand up here is still going to be bound to the serine. So the next step is you're going to have a water. It's just going to come in and it's going to bind to the enzyme right here. And then this water is going to break into the hydrogen here, is going to bind to this nitrogen on histidine. And then the OH here is going to bind to this carbon right here on the fragment of the polypeptide chain.
Mm-hmm. And one thing I just wanted to add with the, sorry, you're not done yet. I apologize. Go ahead.
No, sorry.
Yeah, well, one thing I wanted to add is that we can be reminded that this is, I'll give you guys 5 seconds, a ping pong mechanism. That's right. We start off with our enzyme, the substrate comes in, ES1, and then boom, the product is made, it's cleaved off, and then we're back to a new transformed enzyme state. And then another substrate comes, ES2, boom, product 2 is gone, and now we're back to our original one, which we're about to see right now. John, you gotta keep going. I'm sorry, I didn't mean to interrupt.
Yeah, thanks for that, Bennett. And so, yeah, so at the end, what we're going to have is this hydrogen that is bound to the nitrogen on histidine. It's going to go and it's going to come up here and bind back up to originally where it was on the serine. And when that happens, this bond right here between the oxygen on serine and the carbon on this polypeptide chain is going to cleave. And now you're going to have a second free fragment of a polypeptide strand. All right,
Cool.
Ben, you want to talk about?
Thank you for that. I didn't mean to interrupt. I'm sorry. But again, we're going to talk about the things that I like, which is the predicted effect and how we can use this in the real world. And so for that, we're just going to be thinking about the pancreas and what's causing pancreatitis. And specifically, what does cause pancreatitis? It's the buildup of trypsin in the pancreas and the buildup of all the cascade of digestive enzymes. There's so many digestive enzymes. There are. And the pancreas holds a lot of trypsinogen, which is a precursor for trypsin, which is what starts the digestive cascade in our body. So one thing that we know is that trypsin and trypsinogen can auto-activate each other to create more trypsin, which is, again, the precursor becoming the activated form, which can cause pancreatitis because it doesn't need it. It doesn't need it. There's nothing that can really stop it. But the question becomes, you want to go to the next slide, John?
Yeah.
The question becomes, well, trypsin and trypsinogen already have a natural inhibitor, a natural biochemical inhibitor. Trypsin has an aspartate on its surface, and trypsinogen also has a modified aspartate. And so those negative bonds will repel each other. And so what is causing the aspartate, what is causing the trypsin and trypsinogen to auto-activate each other if they're continuing to repel, and there's no way that they can keep going? And so that's what most scientists are going to be asking. What is the enzyme that's cleaving off for trypsinogen? What is cleaving off the phenylalanine aspartate peptide bond that is responsible for repelling the two, being the natural biochemical inhibitor that causes pancreatitis? Great question. But guess what? We have been able to, we think we found the answer to it, because again, phenylalanine, an aromatic compound, aromatic amino acid, would fit perfectly into our hydrophobic pocket of chymotrypsin-C. And then we also have our aspartate as one of our active sites too. It might be a potential reason too to attract it over. But yeah, that's why we predict that chymotrypsin-C is responsible for activating trypsinogen and in turn increasing trypsin activity in the pancreas. And our results are going to show something similar to that. But before we move on, I just want to remind you guys that trypsin activity is very synonymous to trypsinogen activity, because as trypsinogen activity increases, trypsin activity increases. So if chymotrypsin-C, so we'll just go to the results. I'm going to let John take that over.
Yeah, so in 2006, actually, it was a study by Niboda and Sahintot, and they really studied this. And what they found is, you can see here with the control and all these other 2 enzymes, the trypsin activity is not going relatively fast at all. But then as soon as you put in the CTRC, which is Chymotrypsin-C, boom, it shoots up super fast. It's going almost 2-3 times as fast of trypsin activity as opposed to if there isn't any chymotrypsin. And so what we think is because it's going so fast, it might actually be overactivating the trypsinogen, which is what would allow for pancreatitis to happen. Because it's good to have your reactions going fast. When you have too much, then that can not be good, especially when you're talking about in the body and your digestive tract.
Yeah, and so that can even go into our real day life in medicine and pharmaceutical companies. What can we do to stop chronic pancreatitis? Maybe the answer is in Chymotrypsin-C. Maybe we have to figure out a way to block Chymotrypsin-C from being activated by trypsin. Maybe we have to figure out a way to lower the activity of Chymotrypsin-C so that trypsin activity can go down and pancreatitis can also decrease. And again, it's crazy that phenylalanine aspartate bond is cleaved and it automatically wants to the trypsinogen wants to auto activate with the trypsin and make more trypsin, which we don't even need. And so if we can figure out a way to stop the Chymotrypsin-C from being present or just not allow that to be active, then we will only be creating trypsinogen or trypsin when we actually need it with our normal enzymatic digestive cascade. Just crazy what we can do with such a simple topic, not a simple topic, such a down-to-earth kind of topic. John's going to talk to us a little bit more about the evolutionary side of Chymotrypsin-C as well.
Yeah, so one of the really cool bioinformatics tools that we have now in this 21st century is called BLAST. And so what this does is it can take a DNA sequence and it compares it and says, okay, what other sequences in other species are really, really similar to this. And as you can see here, let me move this over here. Pan paniscus is actually the pygmy chimpanzee, and they have an enzyme, peptidase S1 domain containing protein, that its DNA sequence is 98.9% similar to that of Chymotrypsin-C. I mean, that's crazy. That's like, what, 98.9%? That's like less than 2 bases per 100 bases are going to be different in chymotrypsin compared to this protein that is in a chimpanzee. And all these other ones down here, you see of gorillas, a whole bunch of other different types of monkeys, and just other species in general all have 90% similar DNA sequence. And so it's just, if you don't believe in evolution, I don't see how you can't. Like, it's so evolutionarily similar. And so it's just really cool seeing how there's similar sequences in different species. And so now, Bennet, do you want to tell us a little bit about PCR?
Yeah, dude, I'm so excited, man. I hope that you guys know what PCR is. We took a test on it, and we had it was a lot of points, but I'm sure that you guys already know. But I'm gonna refresh your memory. Sorry. So PCR, we start off with our normal DNA, and in order to amplify it, they're 2 separate strands, and we add a forward and reverse primer so that our DNA polymerase knows when to start and when to end. And if it doesn't know when to start or end, it's not one or the other. If it doesn't know when to end or it doesn't know when to start, it won't do it. And so to anneal, we are going to cool the DNA sequence so that the primers can bind and anneal to the DNA sequence. And then we heat it up again and add Taq DNA polymerase, which is the only thing that can really start growing, and add our DNTPs, which is our nucleotides A, T, G, and C. And we add those in, and guess what? We're gonna have 4 new strands, one from the old, one for each of the old strands. And then we do it again, we have 8, and then we do it again, we have 16, and we do it again, and we have 32, and just like that, we have a ton of DNA sequences we can analyze.
Yeah.
And John's gonna explain a little bit about how we got to this spot.
So that's really cool. With the primers, you can see the first forward primers in the beginning, just the first 20 bases of our sequence. So you can see here. In the second one, you're going to take the last 20 bases, but you're actually going to find the reverse complement of it. So that's really cool. And then, so here are our references, and just want to thank you for watching, and we hope you learned something.
Yeah, so happy and so honored to be able to do this.